You're listening to Medscape's In Discussion series on myelofibrosis, a podcast where thought leaders and clinical experts share their diverse insights and practical ideas for optimizing patient care. This series is hosted by Dr. Tania Jain. Dr. Jain is an associate professor of oncology at the Johns Hopkins University School of Medicine, where myeloproliferative neoplasms are the focus of her research. And she is also the director of the Adult CAR T-cell therapy program for hematological malignancies at the Sidney Kimmel Comprehensive Cancer Center. Relevant disclosures can be found with the episode show notes on Medscape.com or the Medscape app. The topics and discussions are planned, produced, and reviewed independently of advertisers. This podcast is intended only for U.S. healthcare professionals.
Hello, everyone, and welcome to this myelofibrosis podcast series. The topic for today is clinical trials and investigational therapeutics in myeloproliferative neoplasms. And our guest in the house is Dr. Gerds, who is Associate Professor of Medicine at the Cleveland Clinic, and also is very well known for his work, including serving as principal investigator for several clinical trials in the treatment of MPNs and very focused efforts on developing novel therapies in this space. Welcome, Dr. Gerds.
Thank you so much, Dr. Jain. It's a pleasure to be with you here today and with everybody out there listening.
Wonderful. As I was chat GPTing your name, right, because that's what we do now. We don't Google people anymore. It reminded me that you were also the editor-in-chief of hematology of the ASH newsletter.
Yes, ASH Clinical News.
ASH Clinical News, I'm sorry. So how was that experience? I was curious.
Oh, that's really a fantastic question. You know, done a couple of different things within ASH in terms of editorial work. I also did ASH News Daily, and at first you're like, well, we're just kind of reporting things that are going on, but it's really a neat way to engage with the hematology community, right? And you know, it makes me think about things that I never would normally think of. Normally in my everyday work, I would never think about coagulopathies or bleeding diathesis or, you know, thrombosis outside of MPN-related thrombosis, right? So it's kind of a neat way to think about what science is going on in hematology, really kind of get that breadth and really engage with the community, saying, you know, what topics are hot outside my personal area of interest? What political action should we be taking in terms of hematology-related issues? How can we engage with different types of care providers? How can we engage with patients? How can we engage with industry and other facets of hematology? So it's really a neat experience to think about hematology in a very global sense. Not global as in world, but global as in world, but also like the greater community of hematology. And, you know, you get to exercise your creative juices and think about things you normally wouldn't think about. So it's been a wonderful experience.
Absolutely. Sounds like a very comprehensive and kind of out of the box thinking about hematology kind of experience.
Absolutely.
Cool. So we'll get rolling with our topic today. It feels almost like myelofibrosis or MPNs are following some of the trends that multiple myeloma set up a few years ago in terms of having a nice full armamentarium, at least in the context of clinical trials and investigational agents. We have pelabresib and other BET inhibitors. We have navitoclax, the BCL-XL. We have navtemadlin, the MDM2 inhibitor, selinexor, LSD inhibitors, and others that I'm sure I'm not remembering right now. But I wanted to get your sense, like what's exciting in all of this for you from where you look at these things for your patients that you treat in clinic?
You kind of hit the topic right on the head. You know, if you can't be CML, right, where you have an amazing drug like imatinib that cures people with just a pill, you want to be like myeloma, where there's just a laundry list of available therapeutics to use, right, and the different combinations and different manners and all these types of things, where you turn chronic disease into even more chronic disease, right? And I think we're on that pathway, as you mentioned. Not too long ago, we had 0 drugs approved for myelofibrosis, really 0 active therapies. And now we have 4 drugs approved, which is just totally remarkable in my mind, compared to other diseases like AML. You know, AML has made progress, but, you know, in terms of numbers of approved therapeutics, maybe, you know, isn't quite moving as fast as myelofibrosis is right now. So I think that's pretty exciting for what was somewhat an obscure and a somewhat ignored disease for a long time. So certainly absolutely exciting. And you also kind of touched the next wave of things, right? So we had the big JAK inhibitor wave kind of come through, and now we're on this next wave of attacking pathways that aren't JAK-STAT. And really that is an incredibly exciting process, because this isn't CML, right? You block JAK-STAT, the disease still goes on. And so we got to hit it from a couple of different angles in order to really and durably improve outcomes.
It also feels like just translating a lot of the science over the last decade into actual clinically relevant therapeutic agents. The JAK inhibitors have their role, and obviously that's solid. But all these other pathways that can be targeted, as you said, on top of it or instead of it in some cases to bring about clinical benefit. Which brings us to where it seems like most or all of the products are headed, whether to use them as a single agent after a JAK inhibitor, so sequential, versus hitting with doublets and, who knows, maybe triplets in the future up front, which is what the myeloma world looked like over the last 5 or 7 years. So what are your thoughts about that, about the doublet frontline clinical trials, and how do you see them translating into your clinical practice if, obviously, they were to become commercially available?
Well, I think you again are hitting on such critical points here. I think we look back at the last 10 years worth of single agent studies of drugs that aren't JAK inhibitors. We see biologic effects for sure. Proof of that, the research showing that these pathways are important is there biologically in the correlative samples and some of the responses. But if you're not hitting JAK stat, people generally feel pretty terrible, right? The cytokine symptoms can persist. The splenomegaly can persist. And you may not have enough time for that new therapeutic to do what it can do in a very deep sense against the disease. So naturally, since JAK inhibitors are well tolerated with good side effect profiles and the like, naturally you would say, okay, well, we can use a JAK inhibitor to continue to hit the cytokines and the spleen size and really kind of continue to hold that down, while this other drug can come in and do things on a more deeper and biological way to eliminate disease. And that combination makes sense, at least in an worst case scenario, in an additive manner. And then there are various bits of preclinical data that say that these two approaches can be somewhat synergistic and multiply the effect of each individual drug. So combinations, we're not just doing combinations because we're oncologists, right? When we got one drug and two drugs and we put them together. It is somewhat empiric, but not completely empiric. There's a rationale behind it. But it really makes sense because JAK inhibitors aren't perfect, right? So if we look, the seminal comfort data, right? Median duration of treatment, 3.2 years. And if we look at real world data, it's much shorter. The median duration is somewhere, depending on what analysis you look at, ranging from 8 months to 18 months with a JAK inhibitor. So that's a lot of benefit, but not an indefinite amount of benefit. And so therapies that can extend that benefit longer, to me, are going to be incredibly important. We know that patients whose spleens shrink on therapy live longer. We know that patients' anemia, when it gets better, they live longer. And so if we can extend these benefits longer with combination therapy without increasing toxicity too much, we're definitely going to make a big impact. Just JAK inhibitors alone, not to keep looking backward, but if you look at approval, so before ruxolitinib's approval, there was a median survival for people diagnosed with myelofibrosis. It went up dramatically after the approval of ruxolitinib. The average survival for just all people with myelofibrosis, whether they got JAK inhibitor or not. So that's really important too. But JAK inhibitors really do improve survival by doing these things, not by killing off myelofibrosis cells, but certainly making people better. And at a bare minimum, if we can extend these things further, people are going to live even longer and live even better. And then the holy grail, dare I say a dirty word in myelofibrosis, but disease modification, we'll be able to do even better than that. So I think there's plenty of rationale. And of course, we're all very, very excited about the manifest and transform data that was presented at the American Society of Hematology meeting back in December of last year.
You hit on a word that I cannot let go now since you brought it out, disease modifying, right? So
yeah,
that term has changed its color, its meaning in the context of myelofibrosis over the past decade or decades. Where do you think, in your mind, what's the realistic definition of disease modifying using the tools we have at hand in the context of myelofibrosis?
Yeah, it almost gets philosophical at some point, right? Because truly disease modification is just living longer than you would if you didn't have that intervention. So by that standard, ruxolitinib and other JAK inhibitors would be disease modifying. We kind of use it now, I think, as a shorthand for killing off myelofibrosis cells. How do we measure that? Well, we measure it by changes in allele burden, because that's the best thing we have to measure that, best tool we have, or changes in fibrosis score. Those are going to be your kind of two common ways to quote unquote evaluate disease modification. Now, we all, I think, generally agree that these are not perfect. They don't perfectly correlate with improvements in survival, improvement in symptoms, improvement in splenomegaly, these other kind of key endpoints that we often look at as well. And so we really got to, in my opinion, find better markers of disease modification, if you will, especially changes of fibrosis score. For one, it's subjective, but you can get a panel of people to agree or disagree on the grade and kind of work through that. But it doesn't have a lot of granularity to it, right? It's just 4 points. You get a score of 0, 1, 2, 3. And a change of grade 1, does that really mean anything? I don't know. And then lastly, the fibrosis isn't the disease. And honestly, we're trying to look at a reflection of the thing that we're trying to measure, and it's passed through all these filters. Are we truly measuring what we're trying to measure? So I think, there are some really interesting and cool ways to measure disease modification, like this megakaryocyte density thing that Dr. Joe Scandura does, that I think is really fascinating. There are many others. I could be here all day talking about them. I think we need to, in concert with these new drugs, develop better ways of how we measure the impact on the disease.
I agree. I think that's kind of one of the things that our efforts could be focusing now to identify which out of these, as you said, truly correlates with overall survival, which is the ultimate thing to think about in the context of disease modifying. So you also mentioned Manifest and Transform that were presented at ASH 2023, two of the much awaited, and there's others obviously, but we'll focus on those two because those were similar in the context of being first line doublet versus single agent therapy with pelabresib or navitoclax, and had in some ways similar, but in some ways different results. What are your thoughts about those two particular trials? Where do things stand in your mind for them?
These two trials hit on so many hot topics right now. Literally, you could spend hours talking about it, but I'll try not to for the purposes of this podcast. First and foremost, they both were positive studies, right? They hit on their primary endpoints, period, right? They hit on their primary endpoints, period. That's a big deal. We had two prospective randomized blinded trials that were positive for the primary endpoint. That's amazing. And I think we all need to recognize that and not belittle this and get stuck in a bunch of details when these are two positive trials. I think that's really been the major challenge right now, is that everyone's going in the second and third line types of endpoints and ignoring the fact that both trials were positive. In both studies, the combination of Rux plus pelabresib or Rux plus navitoclax did better in spleen volume reduction at week 24 than Rux alone. I think that is so incredibly important to note because of all the circumstantial evidence we have that reducing spleen size can impact longevity and quality of life and all these other really important outcomes. Both are positive trials. Now, I think you mentioned these kind of subtle differences. The populations of patients were somewhat different on these two trials, which I think is important to note. The key secondary endpoints, particularly symptom burden improvement. Neither trial hit on that secondary endpoint, right? With a clinical trial, you only get to answer one question, answer the question, the answer was yes. But the second question that we kind of add in, the bonus question, was a no on both trials. There was a numeric improvement in symptom burden with pelabresib combination, but there wasn't with the navitoclax. But I would also say symptom burden was not worse with the combination, right? So when you have one drug versus two drugs, you would suspect that the two drugs would be more toxic, having more side effects, possibly worsening symptoms or worsening quality of life versus one drug, but they didn't, right? So navitoclax was very similar in its symptom burden in the two study arms, and pelabresib, again, had a numeric improvement in symptom burden. So I think that's so incredibly important, especially if you look at the Transform data. The combination of Rux plus navitoclax did not make people worse. It shrunk more spleens and did not make people worse in how they felt. That is so important. That's what I see when I see those two endpoints. Now, you had mentioned earlier about we need to get away from these endpoints. Well, there is a movement away from these endpoints, and I definitely am in favor of that. I mean, these endpoints are great. We should always measure them. They're historic. I think, they do correlate with people doing better in general, whether we're talking in terms of quality of life or survival. But there's so much more to this, right? There's treatment durability. There's obviously overall survival, anemia improvement, as we saw with pelabresib, and then these other markers of quote unquote disease modification we touched on earlier. So I think there's so many other key secondary endpoints that it lessens the importance of symptom burden improvement when Rux already does a great job. So if Rux is such an amazing drug at improving symptoms, why are we trying to do better? I mean, we always want to do better, but is it realistic to want to do better than that? And saying that, well, this wasn't a successful trial just because the symptoms didn't make it a secondary endpoint, I think is something we should stop and really think about critically before we move on to that conclusion.
Yeah, I agree. I think I see this as investigators or scientists or clinicians. We're not always thinking as a regulatory agency, right?
Yeah.
I think that is where the conflict is not the right word, but that is kind of where the discrepancy in thinking is right now. And I wonder if this is something worth presenting to our regulatory bodies as clinicians who take care of patients, as investigators who design trials with realistic, feasible endpoints.
Yeah.
So if you were to design an ideal trial in your mind, if that exists,
yeah.
we live in a, let's say we're in a parallel universe, what kind of endpoints would you be looking at in terms of designing your trial with, and also trying to convince regulatory agencies to say, this is what I think is most important?
Yes, if I was a benevolent overlord of Galaxy 298 or something. Yeah, I think, it's a conversation, right? And change is not going to happen overnight. So I think it's really on us and the MPN community to continuously go to the regulatory authorities and say, look, this is what's important now. This is what was important then. This is what's important now. And really kind of keep emphasizing these points over and over and over again. There was a kind of this listening forum that the FDA had a year or two ago where they brought in patients, they brought in MPNs experts, and everybody sat down and said, what is important? I think we need to continue to encourage to do that and collaborate with the FDA and other regulatory authorities. Yeah, that is clearly the only way forward here. If I had all the authority in the world to design trials, I think there are some really interesting designs. You brought up something very early on in the podcast about monotherapy versus combination therapy, particularly in the upfront setting, right? We all have tons of patients who do very well on single agent JAK inhibitor for a very long time. And why would we risk toxicity or side effects or other issues by using combination therapy, or costs even, right? So 2 drugs is probably going to be more expensive than 1 drug. So how do we understand that? There's some cool trial designs out there right now. So there's an MDM2 inhibitor that's in development, navtemadlin, and there's a clinical trial that has a really interesting design where everybody goes on standard of care ruxolitinib, and if patients do great, they continue on the Rux. But if they're not hitting critical markers at certain time points at 3 and 6 months, they go on to combination, right? And that's a really pragmatic way of going about things. And that's how we do things in everyday practice, right? If your listeners are not familiar, there's this model called the RR6 model, which I think is a very simple way to think about this. At 3 and 6 months, are you on Rux 20 twice daily? Has your spleen shrunk by 30% in volume? Best you can tell by palpation. Are you transfusion dependent, right? If things are all positive for those 3 markers, you're doing great and you're going to do great on ruxolitinib. But if you're not there, then you should think about alternative therapy. So that's how we already think about things in practice. And so this trial kind of matches that. And so I really like that trial design. Other trials that are ongoing that I think should be models, quite simply, the Entellostat trial, where it's a prospective randomized phase 3 trial with Entellostat, and the primary endpoint is overall survival. That's a black and white endpoint. There's no kind of wiggle room there. If patients are living longer, you're making a big impact and you're modifying disease, no questions asked. And I think we need to do more of that. And with the trials that we have, again, looking back at Manifest and Transform, I think we need to exercise patience there to wait for that kind of data to come out. We're so quick to judge on the primary endpoint, the early data, the week 24 data. That's too early. This is not AML. In AML, you get a CR rate in a month or two, and you can kind of say, yep, great, or nope. You also want survival data to back that up, but you get these early answers. We're all Amazon people, right? We all have Amazon accounts, right? We want our package tonight. We want our package by tomorrow morning.
Yeah.
But sometimes you order a package from another place and it takes a week to get to you, and that's okay. And so if we have to be a little more patient with these trials, hold tight on navitoclax, right? Transform data, if we continue to follow it, might show some really great benefit. The phase 2 data was really interesting in terms of disease modification potential. If we just hang tight and watch this thing instead of bailing on it because it didn't hit a secondary endpoint that may not have value anymore anyways in the current era, we would lose a potential therapy, right? I don't want to lose potential therapies that could be beneficial. If we take something all the way to phase 3, there's potential there. And I really think it behooves us, and the onus is on us to follow it through. So I think that's really important too. But we have to have these kind of continuing conversations with the regulatory authorities. We need to continue to move their frame of reference to what care is today for myelofibrosis and not what it was a decade ago. And open, constant communication, building those relationships with regulatory authorities and with industry are going to be so critical to getting these new therapies out there. I know we have some MPN specialists who are now working at the FDA, which is always good news. I think the community looks very favorably upon that development and should hopefully help us move these things forward.
Yeah, I agree. I think those discussions are happening and I think are critical to that end. You mentioned toxicities and maybe the last question we'll talk about is toxicities in terms of financial toxicities, no free rides, right? So when we add a doublet, we add maybe double, maybe less than double, maybe more than double cost up front. Does everybody need that? And how do you think about that in the context of as these agents hopefully will become available in the near future?
Certainly that the cost is going to be additive, no doubt about it. And if we continue on this path, I don't know if our healthcare system can sustain these increasing costs in any type of manner. So we got to be responsible there too. There are so many patients who do well on single agent JAK inhibitor. Do they all need to be on combinations? Probably not. And so even if, say, Transform or Manifest-2 leads to an approval for these drugs in the frontline setting, I still think we can take a approach where you do try single agent for a short period of time. If there's great benefit early on, great, you stick with single agent. But then if not within a certain time period, say 3 months, just to use an example, then you switch to the combination. There's plenty of phase 2 second line data for pelabresib. There's an ongoing second line study for navitoclax, right? In addition to the phase 2 trials. There's plenty of data there. I don't really think you're going to lose much by doing single agent for a month or two just to kind of see how the response is going to go and then going on to combination versus combination from the very, very start. Again, I don't have data to prove that, but I think it's a very practical way to minimize the financial impact of combination therapy.
Yeah, I think that sounds wonderful. And you're right. Like, not everyone needs doublet up front. The challenge is we don't know who are the ones who need a doublet. And if only we had biomarkers to predict who probably will need a doublet. So the only way to do that right now is, like you said, do a trial of JAK inhibitors alone for a month or 2 or 3 before you add on the second agent, because there's not always that rush of fixing things right now. One of the important things to recognize in the context of myelofibrosis as being a chronic leukemia is to recognize that it's a slow evolving disease where you have time to pause and think what your strategy is going to be.
You made some parallels earlier to multiple myeloma, and I would do that again here, right? So we're not putting people in VGPRs or whatever, or remissions with these combinations, as far as we know. And so you do triplet or quadruplet therapy for myeloma to get these people in these deep remissions so they can have this kind of period of time, right, before their disease comes back. This is different, right? We're just kind of pushing back gently almost. We're not putting people in remission. So that approach makes a lot more sense than it would in a multiple myeloma. And so I think that's a big distinction here, too, that I would like to make, and why it's rational to do something like that.
Absolutely. All right. With that, we'll end our podcast. Just to wrap up, we had a great discussion with Dr. Gerds about how to think about the novel agents that are in the pipeline moving up the ladder, and hopefully some of them, or maybe all of them, will become commercially available to our patients at some point. And along the way, we'll learn about who needs a doublet therapy or what is the right strategy to add on a doublet therapy to the JAK inhibitor backbone, which continues to remain an important therapeutic strategy in MPNs. And we will continue to think about endpoints, or what's the best trial design, keeping in mind trying JAK inhibitors alone for some period of time and then adding a second agent on in those who need it versus leaving those who are benefiting on single agent therapy. This was wonderful, Dr. Gerds. I appreciate you taking the time, and we will meet with our audience with our next episode. Thank you very much, everyone.
Thanks for listening to Medscape's In Discussion Myelofibrosis podcast series with our host, Dr. Tania Jain. Be sure to look for more In Discussion episodes wherever you get your podcasts. Check out medscape.com or the Medscape app for show notes, links, and more information on myelofibrosis.